Good morning, everyone. My name is Apphia Lall. I'm an rising senior at John I. Leonard High School in their pre-medicine program. These past couple of weeks, I've been given the opportunity to work as an intern in the Bolton Lab. The Bolton Lab is focused on disorders of neural circuits, more specifically how neural circuits are altered in neuropsychiatric disorders such as autism and schizophrenia. My specific project focused on if TBR1 mutations can cause defective amygdala axonal projections in models of autism. In order to better deconstruct this project, we need to fully explore each aspect of it, starting with autism itself. According to the American Psychological Association, autism spectrum disorder refers to a neurodevelopmental disorder that is characterized by difficulties in social interaction, social communication, and repetitive or stereotyped behaviors. By definition, the symptoms are present early on in development and that affect daily functioning. The term spectrum is used because of the heterogeneity in the presentation and severity of ASD symptoms, as well as in the skills and levels of functioning of those who have ASD. ASD is influenced by genetic factors. It has a heritability of 45 to 65%. This disorder is polygenic, meaning that it is influenced by a multitude of genes rather than just one. One gene that is thought to cause autistic-like traits is TBR1. This gene is expressed in the cerebral cortex, hippocampus, and amygdala. The amygdala is an almond-shaped structure in the brain that is known for processing emotional memories and fearful environmental stimuli. It has a role in triggering the so-called fight or flight response as it sends a distress signal to the hypothalamus. This structure also communicates with the hippocampus and prefrontal cortex, among many other structures. The amygdala is mainly composed of projection neurons. The projections of these neurons are known as axons and dendrites. The axonal projections help to transmit electrical impulses and information to other neurons. Our project focuses on how and if TBR1 mutation affects the axons of amygdala neurons. TBR1 is a transcription factor, meaning that it is a regulatory protein that has the ability to activate or suppress gene expression. On the screen, you can see a schematic of TBR1 showing the known mutations. TBR1 regulates neuronal migration and the axonal projections of neurons in the brain. So why do we use TBR1 haploinsufficient mice as models of autism? Well, TBR1 haploinsufficiency, which means one copy of the gene is deleted, can lead to ASD-like traits, such as impairment in associative memory, impairment in cognitive flexibility, communication deficits, and social interaction deficits. Most of these behaviors are all highly regulated by the amygdala. TBR1 haploinsufficient and wild-type mice were used as models throughout this project. Wild-type mice did not have any mutations in the TBR1 gene. Therefore, they were the control in this experiment. A paper published in Nature Neuroscience by Huang et al. Noticed that when comparing images of wild-type and TBR1 haploinsufficient mice, the posterior part of the anterior commissure were missing in all analyzed TBR1 haploinsufficient mice. This is shown on the screen as the anterior commissure is highlighted in the wild-type mice, but is not found in the TBR1 haploinsufficient mice. These findings suggest that the posterior part of the anterior commissure is the anatomical structure most sensitive to TBR1 haploinsufficiency. Because the posterior part of the anterior commissure links the two amygdalae in the two hemispheres together, this suggests that interamygdalar connection is impaired in TBR1 haploinsufficient brains. Knowing what we do about autism and TBR1, we hypothesize that TBR1 mutations will affect the amygdala and axonal projections. More specifically, we think that the loss of one TBR1 allele is sufficient to induce axon deficits in amygdalar neurons. Therefore, the goal of this project is to better understand how amygdalar pathways are altered in ASD mouse model brains. In order to accomplish our set goal, we chose to do anterior grade and retrograde tracing injections. These methods are widely used among the neuroscience community. The TBR1 haploinsufficient and wild-type mice are first injected with a genetic material that encodes for the fluorescent marker GFP and M-cherry. For the anterior grade tracing, injections move from the cell bodies to the terminals of their axons. These injections are done in the amygdala, which then travel to areas of the brain that interact highly with this structure. Retrograde tracing injections moves in the opposite manner, from the terminals of the axons to the cell bodies. These injections are done in the prefrontal cortex, insular cortex, and nucleus accumbens and ventral hippocampus. Both of these injection methods are used to view the pathways to and from the amygdala. After the injections are completed, we then wait 3 weeks to ensure that the brightness of the label is sufficient. After that time period, the mice are then perfused and the brains are taken out. The left or right hemisphere are carefully cut in order to ensure accuracy of direction when placing on slides. The brains are then taken to the vibrosome, which is a machine that cuts extremely thin slices of brain tissue. These slices are then meticulously mounted onto clear slides, with approximately 8 slices per slide. The slides are then taken to the 980 confocal microscope to image. Through this process, we can see the fluorescent protein that we injected 3 weeks ago and how it has traveled throughout the tissue. These images are then analyzed on the computer using a program called ImageJ. This is done to view differences in brightness intensity and cell count. These are some of the images that we took using the 980 confocal microscope. In the first image of the prefrontal cortex, you can clearly see the pathway of the fluorescent protein and how it has distributed out throughout the tissue. While we do not have the final results from these images, if our hypothesis is proven correct, we expect to see that the wild-type mice show a higher brightness intensity and cell count when compared to the TBR1 haploinsufficient mice. These are some more images we have taken of the nucleus accumbens and amygdala. Here you can see the fluorescent protein that was delivered to these specific sites. As of right now, we are currently preparing 4 brains to do a light sheet. This process is fairly complex as the brains are placed in a buffer solution to delipidate the brains, which means to remove the fat or lipids from the brain tissue. On the screen, you can see an image of a mouse brain before and after delipidation. The buffer solution is then switched out every 24 hours for 2 weeks. After that time period, we would then use the light sheet microscope to image the brains. This is done in order to confirm the findings from the 980 confocal microscope, and this also helps to provide a full overview of the whole brain for future reference. Another major part of our methodology was electrophysiology. This process is done in order to observe and study the ionic currents in living cells, in this specific case, neurons. I had the honor to work with Dr. Bolton as she showed me around the electrophysiology rig and how to prepare the solutions needed for the process. Through this experience, I was able to grasp how time-sensitive certain research processes are, as the cells are only viable in the rig for 6 to 8 hours. I was actually able to patch a cell and through this I realized how intricate this process is. At this time, we do not have the final results from this. However, if our hypothesis is proven correct, we would expect to see there would be less electrical activity in TBR1 haploinsufficient mice when compared to wild-type mice. As I stated earlier, we are currently preparing 4 brains to do a light sheet. As for what's next, we would continue on with our current procedure in order to get the sufficient amount of data needed to form a final conclusion. Lastly, I would like to express my gratitude for this opportunity and the amazing people I met along the way. I want to give a special thanks to my mentor, Caitlin, and I would also like to thank the rest of the Bolton Lab, who all took the time to share their knowledge with me. I would like to express my gratitude to Ilaria, Erica, and my fellow interns for making my time here very enjoyable, and lastly, the MPFI supporters who have given me the opportunity to be here today.